protein generative model via partially latent flow matching. Thomas and Karen are both research scientists from NVIDIA, and you can take it from here.
Cool. Yeah, thanks a lot. Hi, everyone. I'm Kieran. It's Thomas.
I'm Thomas. Nice meeting everyone.
Yeah, and today we're going to talk about one of the projects we've been recently involved in, La-Proteina. It's kind of an extension of our previous Proteina talk. I think Carsten gave this talk last time, I think, for this series. So yeah, excited to be back. And I think Thomas will get started with like intro, some of the method stuff, and then I will take us through results and some discussion. Yeah.
Sounds good. And everybody, like as we speak, feel free to ask questions if something is not clear or anything comes up. Yeah, so we'll be talking about La-Proteina, our latest paper for atomistic protein design. And Kira and I will be presenting, but this was a pretty big team effort by us, Songlin, Danny, Subai, Chris, Emine, Karsten, and also Arash. And the paper is on arXiv. We have a pretty nice project page with pretty proteins, and the code will open source the code. We're working on it. Hopefully in the next month or so, the code will be out there for people to use. What is this? Okay, yeah. So we'll start with a very quick summary for the whole thing, and then we'll go into the details of what's happening. So we developed La-Proteina, which is a generative model for atomistic protein generation. So we want to produce proteins, complete proteins, the sequence, the backbone, and the side chain coordinates, everything. And the main contribution here, you could say that's the partially latent representation we use for proteins. Because the sequence is discrete, the side chains are kind of tricky to model because they depend on the amino acid type. We basically build this partially latent representation to unify everything into a continuous space. So in this partially latent representation for proteins, we model the alpha carbons explicitly, and then everything else, the sequence and the side chain coordinates, and the other backbone atoms that are not the alpha carbons, are modeled through continuous fixed size per residue latent variables. So once we have this representation for proteins, we can easily define a generative model using this partially latent representation, and we can take advantage of continuous space diffusion models and flow matching without having to worry about this variable dimensionality or the discrete components of the sequence and these things. For our architecture, we use non-equivariant transformers. These are pretty much taken straight from our previous paper, Proteina. They are quite simple and very scalable. We train on a few subsets of the AlphaFold database. And we also show that with our model, we can do atomistic motif scaffolding in several setups. So when you give a subset of atoms as conditioning information to your model and their coordinates, our model can produce proteins that contain this substructure. And this structure can be provided in an index way, so you tell which atoms in which residue they are placed, or in an unindexed way. So Kieran will discuss more in the results section. So yeah. So what are our results? We have state-of-the-art in co-designability and diversity for unconditional generation, state-of-the-art atomistic motif scaffolding. We are actually quite scalable, so we go up to 800 residues, and we still produce co-designable proteins at those lengths. And at those lengths, actually, most baselines collapse to producing no co-designable proteins. And we actually have a pretty nice analysis of the structural validity of the samples we produce. And we show that it's actually, we outperform the baselines in that as well. So to the right of the slide, we have a bunch of proteins produced by our model. So at different lengths, starting from 100 residues, going all the way to 800 residues. And yeah, and this is an example of atomistic motif scaffolding. So we provide the atomistic motif in the lower part of the figure. And as you can see, our model is able to produce proteins that contain this motif. And it's actually able to sample very diverse proteins, all of which contain the motif. And of course, all of these proteins are fully atomistic. We just don't draw the side chains to make it a bit simpler to visualize. So this is the agenda for the talk a little bit. I'll go with the introduction motivation, then what are the main challenges for atomistic design. I'll discuss in quite a bit of detail the partially latent representation we use in La-Proteina, then training, sampling, the data sets we use and the architecture. And then I'll hand over to Kieran, who will talk a bit about the results and the conclusions. And this will include unconditional co-design experiments, generating long-length proteins, this structural analysis that I was just mentioning, the scaffolding results, and the speed of our method, which is actually fairly fast. With that, let's start with a bit of motivation. So as we said before, proteins are a sequence of amino acids. Each amino acid can take one out of 20 possible values. So it's a discrete variable, the amino acid type. And each amino acid has a bunch of atoms. Some of them are present in all amino acids, so the backbone atoms are present for every amino acid type. But the side chain atoms depend on the amino acid type. So basically, for different amino acids, each residue has a different number of atoms. When you put all of these residues together, this chain folds into a 3D structure. And turns out the 3D structure defines the function of your protein. And the novo protein design, which is what we're doing, is basically designing new proteins that are not found in nature with some specific functionality. And there's many tasks that fall under this umbrella, so you can use this for antibody design, enzyme design, and whatnot. What's critical here is that for all of these tasks, typically the interactions between the protein and any other biological structure involves the side chain. So we need to model these side chains properly. And again, what's the main challenge here? There's two things that we observe. First, you have some discrete variables for the amino acid types and some continuous variables for all the atomic coordinates. And second, we have this variable dimensionality of the side chains. As I said before, depending on the amino acid type, you have different number of atoms in your side chains, right? So these two variables are intertwined. They are connected. So you have to model these things. So what's the current state of affairs in this atomistic design? There's basically two families of methods. On the one hand, we have diffusion models that operate explicitly in the data space. And again, this can be divided in two. First, we have the co-diffusion, the models that we call co-diffusion, that are basically running a discrete diffusion model in parallel to a continuous diffusion model. Some examples are multiflow, pepflow. And what they do is the discrete diffusion model models the sequence, and the continuous diffusion model models the coordinates. Then on the other hand, we have what we call the superposition models, such as Piolatum and Propadel, which rely on a representation that can somewhat model all of the side chains together. For instance, Piolatum, which is a pretty strong method, uses the atom 14 representation. So with this atom 14, you can actually represent any residue type, because any residue type has at most 14 atoms in total, right? But the thing is, for some residue types that have less than 14 atoms, you have some non-existing atoms. So what they do is they fill in with deltas at the C alpha position. And this basically gives you a discrete distribution with these delta distributions, right? And that's not that easy to model. So what we observe is that these discrete and continuous diffusion models struggle to achieve very good performance, and all of these superposition models are not that scalable. On the other hand, we have latent diffusions, and again, this can be divided into two different types, you could say. One, when we train the autoencoder, so we build the latent space ourselves from scratch. These examples here are latent diffusion and omniprot. And on the other hand, you have plate, for instance, that uses the intermediate variables of a folding model, in that case, ESM fold. What we observe for these models is that, like in some cases, for instance, in plate, the autoencoder was not trained for this task, and it's super high dimensional, not that easy to handle. But overall, what we observe is that, like, all of these methods that operate in latent spaces deliver somewhat not very satisfying performance. So ideally, what would we want from an all-atom generative model for proteins? First of all, of course, you want to produce proteins whose sequence and structure are compatible. So when you fold the sequence, you actually recover the structure, the backbone, and the side chains. Then we don't want to reinvent the wheel. We have a lot of methods that are very successful in the task of backbone design. We ask ourselves, how can we leverage this success in the context of all-atom design? We want the method to be scalable, because at the end of the day, we'll want to use this for tasks such as binder design, enzyme design, and whatnot. So these methods have to be able to scale to large complexes. And finally, we want to model, of course, all the distributions properly, the backbone, the side chains, and the sequence. We want to capture the diversity in these spaces as well. And this motivated us to go for this partially latent representation. As I said before, this mixed discrete continuous variables and this variable dimensionality presents a challenge for generative modeling. What we propose is a continuous per-residue fixed size representation for proteins. And as I explained, this is obtained by modeling the alpha carbons explicitly. So the coordinates of the alpha carbons, you model them explicitly. And all other atomistic details are the backbone atom coordinates, side chain coordinates, residue type, are modeled through some continuous fixed dimensional latent variables. So this is trained, this is your typically autoencoded setup. The encoder takes as input a fully atomistic protein and outputs these per-residue fixed size latent variables. And then the decoder takes as input the alpha carbon coordinates. And again, the latent variables are what the encoder reconstructs the full protein. So once you train this autoencoder, you are basically able to define the generative model in this space. And this is kind of nice because, as I said before, this is a continuous space that has a fixed size of number of residues times 11, where this 3 basically here comes from the backbone coordinates, the alpha carbon coordinates, and the latent variables are 8-dimensional. So again, we have a fixed size continuous representation for proteins, which we can use to train our flow matching. And that's exactly what we do. We train flow matching in this partially latent space. And after training, what we do is we have our flow matcher that produces backbone coordinates and the latent variables, and then we feed them through the decoder to actually get the structure we generate. So here we go a bit deeper into the motivation for this partially latent representation. This might be a bit repetitive, but I think it's important to emphasize this quite a bit. So one of the main motivations here is, again, not having to handle explicitly in the core generative model task these mixed discrete and continuous variables and the variable dimensionality of the side chains. All of this is handled by the variation autoencoder. Our flow matching model then operates in this fully continuous space, and we know that these methods work well in these setups. Second, we see this as a very natural extension of backbone design methods. So in our case, it's a pretty straightforward, not straightforward, but like a pretty direct extension of Proteina. We produce the backbone as we did in Proteina, but in parallel, we also produce the latent variables that defines all of the other atomistic details. And we know these backbone design methods actually have a very strong performance. And something we found critical was keeping the alpha carbons or the backbone structure at a coarse level and all other atomistic details separate is critical for good performance. And this is mostly because it enables the use of different schedules at generation time. And we'll talk about this later, but this is critical. When we ablated this, if we don't have this separation, if we encode the full protein into a single latent variable, things don't work nearly as well. And by avoiding explicit modeling of all atoms, we actually get a model that's pretty scalable, and we go up to 800 residues. So this is the main motivation for our partially latent representation that we use in La-Proteina. And very briefly, I'll discuss the autoencoder. This is the same figure we had before, right? The encoder, which is a transformer, outputs these per-residue fixed size latent variables, and the decoder that takes the alpha carbons coordinates and these latent variables and reconstructs the protein. We train this with your typical VAE setup, maximizing the elbow, which is a reconstruction loss, somewhat regularized with a KL divergence term. And we show with this very basic setup, we actually achieve near perfect sequence recovery and a mean all-atom RMSD of 0.1 angstrom. So we are also recovering the structures very, very well. And that's the only thing I say about the autoencoder. Happy to answer questions, but it's a fairly standard setup. We find the flow matching aspect in this partial latent space to be a bit more interesting. And here we'll have a few equations, and this is the only notation you need to know. X in red will denote the alpha carbon coordinates, Z the latent variables. So flow matching typically lends a velocity that transports Gaussian noise into your data distribution. Once you train this velocity, you can actually produce new samples by simulating this ODE. So you start with like Gaussian noise, as we said before, and you simulate this for 1 second, so from t equals 0 to t equals 1, and that should give you samples from the target distribution you trained on. What the heck happened? This, sorry. This is somewhat broken.
Just click further.
Okay, whatever, yeah. Here we are supposed to have a figure that's not showing up for some reason. It's the encoder again. Sorry, the flow matching figure again. So you start from Gaussian noise, you produce your samples from the data distribution, which involve like the C alpha coordinates and the latent variables, and then you feed them through the decoder to get your protein. And how we train this velocity here with a flow matching objective. This is your standard flow matching objective. You regress your velocity against the difference between your clean sample and your noise. Turns out that if you train this way, you recover the velocity that actually models your data distribution. In principle, if you train for long enough with a big enough network, perfectly. There's two issues here. First, this doesn't work very well when you do this naively. And this is because we are producing in this ODE here, the backbone and the latent variables at the same speed. So we are denoising these variables at the same rate. And what we find in practice is that producing the backbone faster than the latent variables actually helps quite a bit in terms of performance. And also this naive sampling using this ODE doesn't work as well either. And some technique typically known as low temperature sampling is kind of critical for good performance, at least to get good metrics. And in this slide that comes up now, I'll explain how we address these two things. So this is what we have, the alpha carbons, the latent variables, the naive ODE sampling. First, we use this separation of schedules. So we define it 2 different times. One that we use to diffuse the coordinates x, and the other one that we use to diffuse the latent variables z. So now our ODE looks like this. X follows this time tx, and z follow the times tz. So we can evolve the 2 ODEs separately from 0 to 1. And in practice, we observe this helps a lot. So this is with using some schedule like this. So as I said before, it's useful to produce the backbone coordinates faster than the latent variables. And here in the plot, you have in the x-axis the simulation progress, and in the y-axis the simulation time between 0 to 1. So 0 represents pure noise, 1 represents clean data, and these curves basically show how fast you denoise the coordinates and the latent variables. And as you see, if you use 400 steps with the schedule that we use in the paper, halfway through the denoising process, the latent variables are denoised only 20%, you could say like that, and the coordinates are denoised up to an 80% level. So we are denoising the coordinates, the backbone coordinates faster than the latent variables, and this helps a lot in practice. Secondly, as I said before, also this naive ODE sampling is not great. So using this connection between the velocity that we learned using flow matching and the score of the intermediate densities in a diffusion model, in case you're familiar with diffusion models flow matching, you can actually very easily prove these 2 variables, the velocity and the score, are related linearly. So we can actually build this SDE to sample the same distribution. The idea here is that for any G, which we call the stochasticity schedule, which controls the amount of noise that you inject at every step, these SDEs and these ODEs define 2 different processes, but they have the same intermediate density. So if you simulate the ODE and the SDE, in principle, ignoring discretization errors, you should get samples from the same distribution. But the main benefit of the SDE is that now we can add these parameters here, right? This gamma x and gamma z. These are typically set to lower than 1, and they downscale the noise that you add at every step. And this is what's typically known as low temperature sampling in this space. And most methods that do backbone design or atom design and whatnot use some variant of low temperature sampling. Sadly, it's a bit hacky, but it actually works fairly well in practice. And it's kind of necessary if you want to get good co-designability values and other metrics as well. Intuitively, what this is doing is kind of pruning the tails of the distribution. But again, this is very hand wavy, very hacky, but it kind of works. And the last slide I want to, on my side, it's a little bit about the architecture and the data we use for training. This whole system may appear to be a bit complex. We have the encoder, the decoder, and the denoiser, right, for flow matching. We train this in 2 stages. So first we train the autoencoder, we freeze that, and then we train the denoiser. And it's interesting to mention that all of these architectures are pretty much the same. They're based on a pretty basic core transformer architecture that we got it from the Proteina model. Essentially what this is a transformer with a pair bias attention injected into the logits through this pair representation. One of the nice things about this architecture is that it's quite scalable, and we optionally add these triangle update layers, which are not scalable, but help performance a bit. But in the results, as you'll see, we'll get state-of-the-art performance without any triangle layers. So our model is super scalable and performs very well. And finally, we train a few different models. First, we train up to 500 residues using 350,000 samples from the FDB. And second, we train a model to produce longer length proteins up to 800 residues using a custom subset of the FDB consisting on roughly 4 million samples. And this is kind of stuff for the method. If there's any questions before we move on to results and empirical evaluation and these things, happy to answer them now. We can discuss them more later. Otherwise, I'll hand over to Kieran and he'll keep going from here.
Sounds good. Any questions? If not, I guess we can go ahead. Sweet. Okay. So, yeah, Thomas talked about the method and introduced nicely what we do. Now we're going to talk a bit about how well it works in different settings. I'm going to start with like unconditional co-design. So as Thomas said, like the whole point of doing a all atom method is that you do co-design. So you design a structure and a sequence. And so we want to look how well does this work. Here are a few results from the paper. It's kind of a big table. So maybe just focus on a few rows. Namely, we have PL atom, which is kind of an atom 14 model and kind of one of our strongest baselines, I'd say. We have La-Proteina here without triangle layers, and we have one La-Proteina data point with triangle layers. And then the lower table is the same again. The metrics to look at here is, first of all, co-designability, which means you have your structure and your generated sequence. You fold your sequence via some folding model, and then you see, okay, how well do these match? The generated sequence structure from a model and the refolded structure. If it's below 2 angstrom, you call it success, and then you say like percentage of how many of these things fulfill this. The second thing is diversity, which basically means, in this case, we take all the successful samples we generated, we cluster them, and then we report how many clusters do we have. So it's kind of a mixture of how good your designability is and how good your diversity is. In the second row, we have novelty, where you basically just compare how big is the maximum TM score to the PDB or the AFDB here in this case. Designability says, okay, we ignore the sequence we generated, we just take the backbone and then we refold with MPNN, either with one MPNN sequence 1 shot or we give it 8 shots and look for the best one. And we look at secondary structure. So the like how much is alpha helices, beta sheets, and the rest is coiled in this classification here. So let's look at the first column here first. So the first part of the table. These are like lengths 100 to 500. And you can see that we kind of clearly outperform all the baselines. And pure atom's like the strongest one, 100 to 500 here, because it's 37% overall. And we are like, without tri-layers, we're at 68, 69, and with tri-layers, we're like 75%. Without tri-layers, we're actually also super strong on the diversity side. And you can see the tri-layers, as Thomas mentioned, like triangle updates and stuff, they give a bit of a boost in the co-designability space as well in the designability below here. But it's not that big. It actually hurts your diversity in this case. And as we'll see later, it hurts your sampling speed by quite a bit. So in practice, like in the end, with other results, we mostly use a non-tri-layer model. Below here, you can look at novelty, where it's kind of pure atom is a bit better than us here, but it's kind of in a similar range, I'd say. And then on designability, we get like pretty strong results. And I think one of the interesting observations here is that designability is still a bit higher than the co-designability, meaning if you take your structures and you MPNN them, they will be on average better than if you just take a generated sequence, better meaning easier refoldable. And so I was interested in that. And so we look at this like a bit more in detail here. where you can see on the x-axis the number of residues, and the y-axis you see again this co-designability or designability success rate. And the fair comparison here is the orange versus the green line, because you look at C alpha and only one shot MPNN. And you can see they start out very similar. So if you look at like 100, 200 residues, which is in practice the length that many people, enzyme designer or like enzyme protein binder designer, are interested in, we're still at like over 90% here, which is pretty good, similar to MPNN. But the longer we go, the kind of the adherence between structure and sequence suffers more than just the backbone designability. Which is kind of interesting. And previous methods have seen it as well, but we kind of closed the gap, especially at short lengths, quite a bit. One thing to keep in mind here is what happens if you do a similar experiment with your ground truth data distribution. So if you take the structures from your data, in our case AFGB, similar things apply with the PDB, and how well are your metrics then looking? In practice, this will look something like this. So your ground truth data is actually way less designable and co-designable than what our models and general models produce. And people have seen this in the past. I think Jeannie was one of the papers that mentioned this already quite early on, that there's like a big gap between what we expect our models to do and what our data distribution looks like. And that's, in our perspective, I think one of the reasons why these things like low temperature sampling are so crucial, because we actually don't want to model our full distribution. We want to model like a subset of this distribution. So that's on this front. So summary, we get pretty good co-designability, and like I think as much as we want, honestly, at these short lengths, and at long lengths, this adherence kind of drops a bit. We can also look at, these are basically more on the structures we generate. We can also look at the sequences and look like what the sequence distribution is. And this is basically like a plot showing like the amino acid frequencies of generated sequences. Blue is like uniprots. Some stats are pulled from the EBI. Orange is the training data, and you can see training data and uniprot are often kind of very similar. MPNN means we take the backbones from our model and then we MPNN them. So we then like generate sequence via MPNN. And you can see that MPNN overrepresents certain residues quite a bit. Like it produces a lot of alanines, a lot of like charged residues here, like glutamate and other ones. Valine is not a charged one, but like one of the branched ones, which also kind of often appears. And if you look at our low temperature sampling La-Proteina, we also bias the distribution, which is expected because we like want to get this hairy, designable, various structures that like the folding models like in a sense. But we have different biases than MPNN, which is kind of interesting. Like MPNN, for example, like overrepresents valine quite a bit, which we don't do. On the other hand, we like leucine, isoleucine apparently quite a bit. And like bulky things like tryptophan are like always quite underrepresented. Whereas if we instead do full temperature sampling, we just disable all these low temperature things. We kind of, our model can very closely recover like our train data distribution of amino acids, but this doesn't give you the 90-95% co-designability results. So again, it's a bit of a question of what you want from your model. In our case, since we're interested in what designers filter for in the end, which is co-designability among other things, we use this low temperature sampling setup. There's something in the chat. Okay, let's have a look. How do you learn the times? I think that's a question for the first part. How do you learn the timescale dtx, dtz, for which you trust the evolution evaluation? If dtx too small, then we'll show the equation works, but don't know when it breaks when dtx too large. Do you basically mean like the step discretization? I think the MOIT, you can maybe shortly unmute yourself. Do you mean like how many steps we basically choose to simulate the OD or SDE? I assume that's what he means.
I'm not 100% sure. I am not allowed to.
I can unmute you now.
Okay, I think you can unmute now.
Yeah, I was wondering about the inference part, right? So we learn during the training process, we are learning the velocity, right? And then during the inference, we use that velocity to generate the equation. I mean the evolution trajectory, right? And usually it's it's a large model. So I was wondering that you want DTX to be slightly larger than you could want to be. Of course, you want to be really safe. You can be small. But if I remember correctly, you were saying that take it as a 1 second. I mean, obviously the time scale is not very physical. So I was just wondering, how do you think about small versus large time scale during inference.
So small versus large, do you mean like how many steps you use to simulate this ODE, basically? Is that what you mean?
I was thinking about the discrete step itself, right? How
Yeah.
large the definition of the discrete step should be.
So the thing is, the times 0 and 1 are a bit arbitrary, right? We could define exactly the same with time between 0 and 7, whatever that's. I think it's more like in how many steps you discretize this.
Yeah.
Right. So
Okay, I see.
use mostly 400, but like the absolute the fact that we go from 0 to 1, it's like arbitrary definitions typically using flow matching. But you could use the time the other times, and that's fine. So the actually absolute size of the dx doesn't really mean that much. It's more like you need to discretize this in a way that you don't introduce that a high discretization error.
Yeah, I think you earlier said 1 second at some point.
I said 1 second because you go from 0 to 1 and it wasn't in the slides. So
But it doesn't mean anything.
It doesn't mean anything. Sorry about that. Yeah.
But extending this question to the number of steps, then I guess if you do too much of diffusion in some sense, right, then you might expect that you might deviate away from the the confidence regime if there is some internal reliability score. So I wonder, like, if you've seen that if you take the inference to too many steps, then you might see that the structure looks very weird or something. I don't know if you've seen such pattern.
I'd say the other way around rather, right? If you go to like very few steps, I would actually like there's a huge literature in diffusion and flow matching on like like few step samplers and 1 step samplers. If you use our model into 1 step, you probably won't get very good structures because the model was trained for this.
You for sure won't get good structures.
For sure won't get good structures. But there are a lot of people working on this in the image and video space. You could do something similar here. We didn't do that because honestly, our model is pretty fast as it is with 400 steps, and we didn't care too much about making it even faster. But you could do similar tricks if you wanted to. I think we're going too many steps. I think
if we go up, I mean, we did up late like number of steps. We just stuck to 400 because it worked well. If we go higher, performance is pretty much very similar. So how we see that is the discretization error being introduced at the 400 steps level is not that significant. If we go lower, like 300 worked as well. If we go to like, I think it was like 100, 150, performance was starting to drop a bit. So going up, the smaller the step, in principle, the better, and performance doesn't get worse. It just stays the same.
Got it. Thank you so much.
Cool. Was there anything else? I think that was it.
There's another question in the chat.
There's another question. Or two of them. Okay. Does this model use proportional MPN for sequence generation and generate structures? No. I mean, I think Thomas explained this, but I can surely repeat. Basically, no, it doesn't use MPNN. In this graph here, we have MPNN just as a comparison. So this green bar here basically means we generate both sequence and structure with our model, but then we throw away the structure in the site to generate. We just use the backbone and use MPNN to generate the sequence. But usually what we do is the red or the purple bar is we generate both sequence, structure, and sidechains at the same time, and then we just use the sequence that our model generated, which in theory should have some advantages, especially if you want to do precise atomic placements, because the model can reason over these side chains and these contacts. So no, we did not use MPNN for sequence generation by default. Next question. Why does including tri-layer triangle updates lose diversity?
We don't have an intuition for that, really, unfortunately. That's something we're quite interested in. What's the effect of these triangle layers? How they help, how they not help. But we don't have a lot of intuitions there yet. Yeah, it's kind of a bit of an open question in the space, I'd say. Like when we started this, people told us quite often oh, this won't work without triangle layers and triangle updates. In the end, turns out to work, and whenever we are at a new task or tackle a new problem, it seems like it doesn't work at the start, and then if you do some tweaks, it kind of starts working. But yeah, it seems like for some people it works quite not without the layers. And it might also, sorry, one more comment. It might depend on the representation. We tried something like the Atom 14 representation, and in that case, without tri-layers, it was quite tricky to get it to work. Yeah. So in that case, tri-layers appear to help a lot more. In our case, for this latent formulation, this partially latent formulation, things work a bit out of the box without the tri-layers. Yeah, it seems pretty robust to architecture choices. Okay. Have you tried fixing the alpha atoms to the ground truth for pure sequence design? What's the performance then compared to protein MPNN?
I think we didn't do that experiment.
not.
Like I think we train sometimes with that, right? We have some training iterations where we do inverse folding or folding, but we don't like just did like a proper comparison between that.
Yeah.
But you could use it for that if you wanted to. I think the nice thing is like MPNN basically, and we didn't do this, but MPNN in theory just allows you to fix backbone, right? Let's say you now want to do sequence design. As part of this, you could also diffuse the backbone a bit again if you wanted to. So it'd be like MPN, but let the backbone adopt a bit as well if you wanted to. What is the reason between training purely on AFDB structures in comparison to, say, a mixture of PDB and AFDB structures? We did this in Proteina, for example, right? I think we did both pure training, we did LoRa, we did kind of mixture of things.
I mean, honestly, this bigger, I mean, we could train on the mixture. We just found that the training on the FDB works right away and gives like
pretty good performance.
Nice metrics, and good performance, produces nice proteins. You could consider training. I mean, training only on the PDB might be a bit tricky with these bigger models. You should explore, like we've been exploring smaller models for the PDB as well that appear to work well. But like the architectures we use here that have like 400 million parameters, on the PDB, they overfit fairly easily and performance is not that great.
Yeah. I think it pretty, as Thomas said, it pretty directly extends from Proteina where we did this and kind of worked. So yeah, for this we were basically, it was like not a data paper in a sense, but you could do this. I think we did this at some point, it worked, but like for the final stuff, we just kept these things. How did you relate dimension of 8 based on VE performance alone, also performance of the flow matching and generation evals?
So this will actually, Kieran will discuss a bit more at the end. The autoencoder is a bit of a sensitive part of the system. But we had autoencoders with latent variables of dimension 4, and it actually performed quite well as well, slightly worse. We had 16, didn't really help that much. It was a bit ablated, but always in terms of training a latent diffusion on top of it. Reconstruction-wise, and these losses for the autoencoder were always very good, like sub 0.5 Armstrong RMSD and sequence recovery pretty much perfect for all dimensions that we tried for 8 and 16.
Yeah, I think that's a general conclusion of the project, I'd say. Kind of like, we talk about more about this later, but the autoencoder reconstruction performance itself doesn't really tell you that much about how well it will perform as a latent space for a diffusion or flow matching model. Are you training your encoder in the autoencoder from scratch or is it pre-trained?
That's from scratch.
Yeah. So the encoder and decoder are joined together, they're trained together from scratch, basically. So we don't use anything like plate where we have like a folding model initialization or something. This is all from scratch. Okay, I think we're caught up with questions. Cool. Let me continue. Okay, so yeah, I mentioned like if we do full temperature sampling, we kind of recover the amino acid distribution of our training samples. If we do low temperature sampling, we bias the distribution a bit, which makes sense. But we don't do some bias that people usually, like for example, MPNN has this alanine bias people often see, and some previous co-designability methods have it as well, and we don't have that. We kind of have like a similar alanine content here as well. So I think overall we're kind of like happy with these distributions, I guess. Yeah, as Thomas mentioned, like our architecture is really pretty scalable, so we can train and also evaluate on quite long lengths. So this is a comparison on the left. The two left plots are backbone performance, and the two right plots are co-design performance. The left plot is always designability here. So designability and this co-designability, and here's like number of designable clusters, and here's number of co-designable clusters. So these and this plot is about for diversity. And we can see on the left one, if you focus on backbone, we have a lot more baselines because there are these previous Proteina here, Proteus, after fusion, we have a lot more baselines, but they all kind of collapse at some point here, around like 600, 700, for sure at 800. Whereas Proteina was, I think, one of the first like that stayed like quite performant until quite long lengths. And we actually improve on this with La-Proteina, meaning like the backbones, all atom modeling helps even your backbones themselves, which is kind of nice, both in designability and diversity. And then if we look at co-designability, where we have a lot less baselines. So basically these three here on top, four here. So plate, property, protein generator, pure atom. They also quickly degrade here at 400, 500, whereas we also degrade, as I showed before, which is not too unexpected given the training distribution. But we still generate like co-designable samples at these lengths, which is kind of nice. Cool. All this eval so far kind of relied on folding models, and we all like our folding models, but we also want to look at like, okay, we now place atomistic sidechains. Can we somehow check independently if like the biophysical context of this actually makes sense? And so we're thinking about how to check the atomistic quality like beyond just refolding. And one thing that you can think about if you look at a PDB entry, this like carbonic anhydrase, I think, what you always have is this like validation report here. So you have like clash score, sidechain outliers, etc. And for each PDB that's publicly uploaded, you can get this full report here. And if you look at this full report, you will see that they use a tool called mod property for this. And this tool basically evaluates your structure against a lot of other reference structures, which are like termed to be of good quality. And then the PDB has some thresholds that you should like observe so that like when you refine a structure based on experimental data, it should have this certain quality. And the things they look at are things like clashes, hydrogen bonds, atom contacts, like rotamer outliers, angle and bond outliers that are weird. And some more like specific things like with Rebo, like in DNA cases or C-beta outliers, etc., etc. So it basically checks for like biophysical quality and how similar is your thing to how usually things look like in the PDB and what we know from biophysics, basically. So what we did is like, okay, that's kind of neat. Let's just like run our structures and all the baselines, all atomistic structures that are generated by our baselines through this tool. And let's like have a look of how will they perform. We have more plots in the appendix of the paper, but these are like the four main ones, I guess. On the y-axis, you also have the score, and of the x-axis, you have the residue length. So from 100 to 800 residues. The first one here is the mod property score, which is a combination of a lot of other scores. So this is kind of an overall quality metric. Then we have the clash score, which basically tells you how many all atom clashes, like van der Waals dissections you have, angle outliers and bond outliers. In red is like a rough from this paper I linked below here. We have some reference values for like high quality X-ray structures. So I just took some of these and like put them as red lines in here. And the green line is where La-Proteina is on different lengths, and then p-all atom is blue. And then you can see the other baselines here, APM from ByteDance, I think, protein generator, and a few others. You can see it consistently like our method performs best across like all physical, like biophysical validity metrics, especially on the angle and bond outlier. We see that other methods have quite a few of these, while ours like stays pretty realistic and below or around the level that high quality PDB structures look like, which is quite nice to see. Another thing we were thinking about there was, well, we have all this low temperature sampling, right? And we have low temperature sampling for both the backbone and the sidechain. For the backbone, we have still diversity, right? Both in terms of amino acid sequence and in terms of structural diversity, as you saw on the table. But what's about the sidechains? Do we always collapse to the same sidechain? And the sidechains in amino acids and proteins generally have specific configurations. They're called rotamers. So some of the angles are rotameric, meaning specific configurations are often observed. And so in our paper, we look at this. We look, okay, how do the different sidechain, like the chi angles, look like for proteins over like a variety of proteins? And then this one example, you can see all of these plots in our paper, but this is like the first chi angle of tryptophan. And you can see in orange dot is the AFDB and in red dots are PDB. They're very similar, which like is expected, I guess. These are a few hundred samples of different lengths. And our model, La-Proteina, kind of nicely traces these three different rotamers here. You can see at minus 90, at plus 40, plus 60, minus 60, and 108 degrees, basically. So you can see like with this three nice rotameric peaks. Some other models here miss these peaks, or some like collapse on a few rotamers and miss others completely. I think there was some. And some others get the right rotamers, but like different distributions, like overrepresent some of them. So we are quite happy to see that we actually, despite the low temperature, kind of follow the distribution here quite nicely. The next thing we looked at was like, okay, we have this latent space now, this per-residue latent space. And the question, like, we train this autoencoder, like, what does this latent space actually represent? And what can we learn from this about how our model might generate these side chains and proteins? As Thomas mentioned, we generate the backbone first, but then when we generate the backbone, how does the model generate things? Does it first generate like sequences and then side chains, which makes sense? Or how does this work basically? And so one way of doing this is looking at like a visualization of the latent space. In this case, like a t-SNE, dimensionality reduction. These are always like a bit in quotation marks because there are a lot of hyperparameters, etc. But this is like, I think, the default config, just like running t-SNE with the default PCA initialization, I think. And you can see like, first of all, the amino acids cluster together. So like these are like some residues from generated samples. So it's nice. We see that glutamates are all together, tyrosines are all together. So that kind of makes sense, I guess. But the other nice thing you can see is that like similar amino acids also kind of cluster together. So you can see on the right here, these are like phenylalanine, tyrosine, histidine, tryptophan. So all the aromatics are here on the right. You can see smaller amino acids like cysteine, alanine, serine, like they're all on the left here. Aspartate, asparagine are close, glutamine, glutamate are close. So structurally similar acids are kind of similar in space. This is by no means perfect, and this is by no means like a perfect visualization, but it kind of gives you an idea of, okay, this latent space behaves healthy and like kind of like we expect it to be. And then the question is like, okay, how does during the noising process, how does this information get destroyed and during the generation also restored? So it's like what this experiment on the right here shows. So on the x-axis here, you can see on the left, you have a clean latent, so from a clean structure. And then the more you go to the right, the more noise you add to it. So basically you go from clean structure to the prior to noise. And then the green curve shows what happens to the structure, so how much the structure deviates. And the blue curve shows how much the sequence deviates. And the like dashed lines here are the residues that perturb, and the like non-dashed lines are the non-perturbed residues. And the first thing to see is like if you perturb one residue, the other residues don't change, which is, I guess, good. So this latent is like very local per residue. And the other thing you see is if you only add some noise, the structure changes, but the sequence stays the same. So you can imagine this like, okay, you're in this glutamate cluster, for example, and you change like where in this cluster you are, but you don't jump out of this cluster. The more noise you add, at some point, you then switch to a different amino acid. So you'll, I don't know, jump from glutamate to glutamine or whatever, some other amino acid here. But going backwards, basically, when you generate, this kind of shows that like you have like some like plausible generation trajectory and some plausible orientation of these amino acids together in this latent space. Okay, yeah, we have some pretty samples, I guess. And with that, we can also do like all atom generation here on the left-hand side, some samples, some Tim barrels, some other ones of like short and long length. You can also do pretty nice motif scaffolding. So here in red, you can see a motif, and then our structure nicely incorporates this motif. And we can not only do this with like full side chains and like full like long segments, but we can also do this for like only like small active sites, as in this case here. We only give like different like histidine rings, histidine tyrosine rings here, and like a carboxy group and like some other, I think it's serine or something. So like a very small atomistic motif to the model. And the model learns to incorporate these into like a variety of backbones and recapitulates these like very well, basically. And like in a beta sheet here, or some very alpha helical protein, and all of these are co-designable in the end, which is quite nice. The cool thing about this was, and we are kind of ourselves, I guess, a bit surprised by this, or not surprised, but we are playing with different ways of incorporating this motif information, because I think one thing people thought about late representation is it's harder to enforce this explicit atomistic constraints because you don't have explicit coordinates anymore. You need to like somehow encode them via the latent space. But just giving these as labels during training, basically just telling the model, okay, I want these 3 positions to be fulfilled in the end, worked out of the box more or less. So we didn't need to do like extra decoder losses or anything. The the model kind of nicely adapted to these tasks, which is quite nice to see. We can look at this a bit more quantitative. So here's like on the left, you can see the performance in all atom motif scaffolding, meaning you have a whole side chain that you scaffold. And on the right, we don't get the whole side chain, but just the tip atoms. And we adopt here this bench or this like notation from Potbadeli, which at this point was the only paper when we published this could do this, or I think it's still the only paper. There's a code available, I think, right now. So it's the only one we could sample. And they have this tip atom benchmark where they say, okay, the last rotatable bond. So the only thing after everything after this, like carboxy group or something, so we scaffold. So in gray, triangle is their success. The green one is our success indexed, meaning you tell the model where in the sequence the motif should be. And then the orange diamond here basically is unindexed. As Thomas mentioned before, unindexed means we don't give the model the information of where in the sequence the motif should be. We just give the 3D coordinates. And the model should itself figure out how to arrange the protein around this. So how much gaps it does between discontiguous motifs, for example. And you can see that like for like 1 and 2 segments, the performance is kind of similar. 1 or 2 segments meaning you either have one chunk of motif or you have 2 separate chunks with some other amino acids in between. But the more segments you have, like 3 or 4 segments, this unindexed performance becomes a lot more important because it gets a lot harder to scaffold this. And the model can then smartly figure out how much amino acids to put in between. And I think Artifusion 2, they also did something like this. And they also showed that like the more islands, I think they call it, we call segments here, the more islands you have, the more this unindexed performance helps you. And we see a similar thing for the tip atom case here. And yeah, I think that was everything on the motif side. One other thing which is kind of nice, I think Thomas mentioned this, because our architecture is quite scalable. That means we can train for and inference quite long proteins, but it also means we can inference like normal or short proteins pretty fast. So this is like a sampling speed comparison. Again, a lot of numbers. You can maybe just focus on these 2 columns, like 100 length and 500 length residues. On the bottom here, we have like 4 baselines of backbone models, like Genie, Artifusion, FrameFlow, and Chroma. Then here we have batch size 1 for a lot of the all atom methods. And then we have maximum batch size. So only, I think, Plate and us allow actually multi-sample, like batched inference. So here you can kind of amortize. I think it was an A100, I think we did this on. So this runtime is a normalized per sample. You can see that like what Artifusion, for example, for 100 length protein takes like 20 or Genie 50 seconds, putting generator even 200. We can do this like around like 3 seconds, and then amortized. If you have maximum batch size, we can actually go like below a second overall. So you can do like very high throughput generation of these short proteins, which is quite nice. And this extends also to longer proteins. So for 500 residues, Latent, for example, already like is out of memory because that is very expressive, but also highly expensive tri-layer operations. Other baselines get very slow, whereas we like escalate like 6 seconds here and 5 seconds with maximum batch size. And at these lengths, you can also clearly see the difference between the tri-layer proteiner and the non-tri-layer proteiner. So I think here, because the performance difference wasn't that big, but the speed difference is quite large actually. We just mostly went with the model that doesn't have the tri-layers. Cool. And before going to conclusions, I think there are a few things in the chat.
Yeah.
Let's have a look at this. Where did we stop? Oh, yeah, break line. Thanks, Leo. That's helpful. Okay, 2 questions. We don't need to do capture alignment for the C alpha coordinates at training time, as similar all atom models do. Have you tried train time rigid alignment?
So we actually did not try that. We know that PLATOM, AF3, they all do that. We did not use it. We don't need it for performance.
Yeah.
At some point we thought about trying it, but like our method was working well.
Other things come up always.
Yeah. And also like, I mean, I may be wrong here, but I don't find it super principled. It might help performance. It might not. In our case at least, we should. I mean, at some point we might give it a go, but we haven't tried it.
Yeah. Second part, how do you make sure or measure that the co-design sequence structure aware of instead of shortcutting in the sequence latent space without paying attention to the structure generation part and vice versa. So you basically mean like how to make sure that the sequence and the structure are coherent. I guess how we measure this is kind of via the refolding, right? If like your sequence would be very different to the backbone you generate, your refolding amnesty would be quite large. That's one issue.
We don't enforce it in practice. We just
Yeah.
train with samples from the joint distribution, right?
Yeah.
So the joint distribution naturally gives you pairs that are connected. So coherent pairs of sequence and backbone, right?
I guess in practice, many like previous co-design methods also train on pairs, right? But it doesn't work sometimes.
I don't know what.
Yeah, well, I guess, yeah, we don't explicitly like have any specific thing for this. I guess this latent formulation just lends itself nicely to encoding this discrete decision into the latent space. And it turns out to work pretty well. But we don't explicitly have specific enforcements for that. How much variability in structure sequence from multiple generations of VAE? Do you remember generations? Like different checkpoints?
No, I guess so.
Oh no, like
This may be different samples from the prior. So from the normal, in a VAE, you have a prior distribution that's a standard Gaussian, right? If you sample the standard Gaussian a bunch of times, you likely won't get very good results, and also you will get very different things.
Yeah, I think he means like multiple generations. Like if you have the same
latents.
Same latents, but we also do like I think he says like the latents is stochastic, as if like the VAE decoding is stochastic.
I mean the VAE, the encoder and the decoder are defined distributions, right?
Yeah.
The decoder we always take the mean. The encoder we do have a Gaussian with some mean and variance, but as we showed before, the reconstruction is pretty much perfect.
Yeah.
So if you take the distribution in the latent space defined by the encoder, regardless of what sample you get, you'll likely get very similar proteins at the output because they are all pretty much very, very close to the original protein that's being encoded.
Yeah, especially because if the C alpha is kind of an anchor in 3D space, that makes things very similar. When you're modeling the joint distribution of sequence, structure, and side chains, what do you see mostly contributes to the structure foldability? Accurate sequence generation as well as diversity of designs. When you in structure foldability, when you just talk about like, okay, we take our backbone structure and we put MPN on it, I think you don't need good sequence generation for this. Like I think there are previous methods that showed like, okay, you can still have like a very good backbone generation. Your sequence can be like pretty garbage. That can happen. I guess in our case, when we properly want to model it jointly, I think as Thomas mentioned, like the decoupling of the generation times was pretty important. So that you have like your model has a rough idea of the backbone first and can then reason about sequence and sidechains. I think that was pretty critical for our success and also for diversity. So that your model has kind of like an idea of what the backbone should look like and can then reason about the finer details.
Yeah.
What kind of augmentation do you use to get around equivariance issues?
We just rotate the input protein, that's it. Random rotation, everything is centered, except not when dealing with motif scaffolding, but in general for unconditional, we center all proteins, and that's pretty much it. And we apply a random rotation every time we sample one from the data set.
Yeah. Okay, maybe just finish shortly with the last slide, basically, and then we can take more questions. We have some additional results. I think we're already on time. These are all in the paper as well, just some ablations. I think the main interesting thing would be like the learning and takeaways. I think we talked about this. We tried to come up with some points yesterday, I guess, of like what did we learn from this project, and what do we think is useful for others maybe as well. I guess first of all, Thomas mentioned this, but like we had pretty good backbone performance to Proteina, and they're also like after fusion and stuff, models that have very good performance. And our partially latent representation kind of builds very strongly on this. So kind of we kept what works and kind of added the necessary complexity that we needed for our latent generation and didn't try to kind of reinvent the wheel, which kind of helped the project. As I just mentioned, separation of these denoising processes between like backbone structure and sequence sidechains was pretty critical. Makes sense. Like people just used to do backbone and then strict sequences then afterwards for a while. So kind of not making it as strict, but emulate this partially kind of helped a lot. The autoencoder checkpoint, I think someone asked this earlier, is pretty critical. I think in different domains people have observed this. That like just because you have good reconstruction loss, that doesn't mean that generation of your diffusion models will work well in this latent space. So ideally, you would have some metric like diffusability or like how nice your latent space to diffuse in. It's pretty hard to quantify or judge this a priori. And I think also in the image space, it's kind of like not a consensus yet. People try a lot of things, equivariant latent spaces, wrapper-like alignments to pre-trained models. But I think there's no consensus yet on how to like quantify this or judge this properly. So I think that's still like an open question. As I mentioned with the atomistic constraints, we were thinking about, okay, do we need to do some tricks in order to like enforce these like precisely in the latent space? But it turns out these are not needed. If we have this, our latent space pretty well adopts to these, and we can use these like out of the box more or less constraints, which makes us hopefully a lot more useful for like other tasks like enzyme design and other things. The triangle updates, again, this project again, people were kind of critical and thought about like, okay, maybe these are quite crucial for performance. Maybe that's like where you hit a wall with the current protein approach, but it seems like it still work quite well. So they're not that crucial for performance, but hurt scalability quite a lot. So you mostly opt to not use them. And here the last point is a bit more like discussion, I guess, like what we want from our models versus what we have in our data. I mean, I showed these sequence distribution plots of like full temperature sampling versus low temperature sampling. If we want to optimize these folding metrics, like we can kind of like tune our models and our temperatures and our schedules to do that. But maybe we also sometimes want to generate things that look more like what's in the PDB or in Uniplot in that case. So I guess this will most likely depend the application you have. But I think it's good to keep in mind that maybe getting 100% co-designability is not the end goal. Yeah, I think that's it. The paper and the product page is Thomas mentions online. We're working on the code. So yeah, thanks for listening. And I guess we're happy to take any more questions if there's some. Thanks.
Well, thank you. If anyone have more questions, you can just unmute yourself.
I can ask one more. Thanks for the great talk. What do you guys think about like, or I guess in terms of conditional generation? I mean, it seems like with all of these latent embeddings and representations, it might make it favorable to kind of like, you know, ideally we could be like, I want a protein that has such and such properties or per-residue properties and this kind of stuff. I imagine you're working on that, or just would like to hear your thoughts.
When you say residue properties, do you mean like this binds to something or like an active enzyme, or it's more like per-residue, something this is like a polar amino acid or something like that?
Yeah, both maybe.
I guess for the like residue-wise ones, I guess like for the coordinates you showed, that's pretty easy to do, right? For the motif scaffolding. I guess let's say you want to enforce that it's like, I showed this latent space, it's like an aromatic amino acid or something that should work pretty well, I would assume. Regarding like more general tasks, I guess it's not a latent space, it's a residue latent space. I think that's to keep in mind. It's not like a general like fixed size protein latent space. So I think if you think about, I don't know, putting like Go labels or something in there, you could do stuff like that. Not quite sure how well that would work, but I guess in Proteina we did this already with the calf labels. So in Proteina we didn't do this in this paper, but I guess we could have done. In Proteina we did this like calf level conditioning, where you have this like C high level class, like is alpha beta is beta, and then you have like lower folds, is like immunoglobulin fold, is Rosman fold, conditioning like this. That worked really well in Proteina, and I wouldn't see why this wouldn't work here. I guess we just didn't want to do it. I didn't have time, no priority, I guess. But these kind of things you can easily enforce. I think it doesn't necessarily depend on the latent space. I think the latent space here is, as Thomas mentioned, quite useful for getting around the like discrete decision and like this like mixed discrete continuous modeling problem. I don't think it necessarily influences your like conditional performance. Cool.
Cool. I guess no more questions.
Yeah, seems like it.
Well, thank you everyone for coming, and thanks Karen and Thomas for your great talk, and we'll see you guys next week for our next seminar. Thank you guys.
Thanks
Thanks a
everybody.
lot.
See ya.
Bye bye.
Bye.